So let's continue our discussion on this chapter as we're talking about microbial metabolism on lecture number 5. Okay, so where do cells get energy? That is the question. So most cells are going to get their energy from this 6-carbon sugar. So 6-carbon sugars are the major source of energy for the cell. When we think about it, what type of macromolecule are 6-carbon sugars?
They're a type of carb?
That's right. They're a type of carbohydrate, right? It's a type of carbohydrate. That is correct. And cells break down this carbohydrate, specifically or usually glucose, to make energy in the form of ATP. Okay, so glucose is that carbohydrate that is mostly used when we're talking about breaking it down and releasing energy. So we're going to be talking about that today. In fact, we're going to be talking about carbohydrate catabolism, also known as cellular respiration. There are some key pathways with cellular respiration that we're going to go into a little bit of detail. And those are glycolysis, the Krebs cycle, and the electron transport chain. Another method or pathway that we're going to be talking about is fermentation. This is another way or another method of breaking down sugar, of breaking down glucose. The difference is, as you can see, one of them has a few more steps, the other one doesn't. One thing that they both have in common is that they both begin with glycolysis. You see that? But fermentation goes from glycolysis straight to fermentation. So when we take a closer look at this, we see that we begin with our 6-carbon sugar. It doesn't matter which pathway you're going to lead. Both of them start with glycolysis. Glycolysis. Glyco meaning this sugar, lysis breaking apart. So we're breaking down this sugar, and in the process, we see that in glycolysis, excuse me, we're going to release ATP. After this first pathway of glycolysis, we see that there is a separation between the cellular respiration pathway and the fermentation pathway. If we continue on cellular respiration, we see that we proceed from glycolysis to the Krebs cycle, in which we release more ATP, and we also reduce carbon dioxide. And then we see that we enter into the electron transport chain, where even more ATP is released. With the fermentation pathway, what we notice is that we start off with glycolysis, right? And we release ATP, correct? Yeah, but then from glycolysis, we proceed to fermentation. And we notice that with fermentation, it says without oxygen. What we also notice looking back is that there is oxygen involved in the electron transport chain. But another observation that I make is that when I'm looking at fermentation, I don't see that any ATP is generated, right? Do you see that? Do you notice that? And then from fermentation, I see that there is some sort of end product of like alcohol or lactic acid. At first glance, I notice that my top pathway releases significantly more ATP than fermentation, correct? Yeah, so we're going to take a closer look at that. So what is the purpose of cellular respiration or carbohydrate catabolism? The purpose is to generate ATP, right? ATP is needed. We just talked about ATP in our question of the day. You mentioned how these anabolic reactions require ATP in order to synthesize larger macromolecules, for example, correct? And it's not only to generate ATP. This pathway also forms intermediates for other synthetic reactions. This is the whole pathway of cellular respiration. It's a beautiful pathway, and we're going to take it step by step. We're going to begin with glycolysis, and we see what is generated in glycolysis. Then we're going to proceed to the Krebs cycle, and then finally the electron transport chain. But before we do that, we need to understand the methods of ATP production, or what are the methods that are used to generate ATP. You need to know these. Now, there are three, but in this class, since this is a prokaryotic class, we will be focusing on just two. So what am I asking? If we're generating or making ATP, through what method? Was the ATP generated through oxidative phosphorylation, or was the ATP generated through substrate level phosphorylation? What does each of these mean? Okay. Energy released during oxidative phosphorylation reactions. Basically, this refers to, well, let me see, what was I going to say? Oxidative phosphorylation. Let me back up. Organisms use three mechanisms of phosphorylation to generate ATP. What does that mean? Remember that from the previous lecture, we talked about ADP. Okay. And then we talked about how we add an inorganic phosphate to ADP to generate ATP. Do you remember that? Okay. That is phosphorylation. Okay. And that is the method that we generate ATP. Now, there are two ways or three ways that this can happen. Organisms use three mechanisms of phosphorylation to generate ATP. Those three mechanisms are oxidative phosphorylation, substrate level phosphorylation, and photophosphorylation. In this class, we're only going to focus on the top two. Yes? Okay. Number one, oxidative phosphorylation. This is the process in which ATP is formed as a result of electron transfer from NADH or FADH2. I know this is probably a little confusing, and maybe it doesn't make any sense, but hopefully by the end of the lecture, it will. Oxidative phosphorylation is the process in which ATP is formed or generated as a result of having NADH or FADH2. Okay, you need to know this. Oxidative phosphorylation is the process in which ATP is formed or generated, right, as a result of electron transfer from NADH or FADH2 all the way to oxygen. Okay, now this transfer of electrons through a series of electron carriers will occur in the electron transport chain, or also known as the ETC. Okay, so this transfer of electrons will occur in the ETC. And it requires NADH and FADH2. The second generation of ATP or method is going to be substrate level phosphorylation, also known as SLP for substrate level phosphorylation. In this generation of ATP, a high energy phosphate is directly transferred to ADP. So we have a high energy phosphate, boom, directly transferred to ADP to generate ATP. In other words, substrate level phosphorylation will generate ATP directly in that pathway, directly. Nothing needs to travel to the electron transport chain. In other words, okay? Nothing needs to transfer to the electron transport chain. Finally, photophosphorylation occurs with photosynthesis. It involves chlorophylls, It also involves the electron transport chain. And in this class, you will not be responsible for photophosphorylation. Okay, so I'm not going to examine you on that. Yeah, all right. Now, when we're talking about oxidative phosphorylation, that it utilizes the electron transport chain, this occurs through a series of oxidation reduction reactions that we're going to be discussing. Okay, so what does it mean to have oxidation reduction reactions? Okay, oxidation reduction reactions, they're always coupled. If something's being reduced, something else is being oxidized. Okay, they're referred to as redox reactions. Something's donating an electron, something is accepting that electron. And as a result, something goes from being oxidized to reduced, okay? That kind of thing. Now, oxidation is going to be, oxidation is going to be the removal of electrons or hydrogens. Reduction is going to be the addition of electrons or hydrogens. I used to spend a lot more time discussing this and giving examples. And students always had a hard time with this. And then eventually I thought, you know what? A lot of you are going into the healthcare industry. It's really not going to be that important that you understand this, okay? And it was so painful for students that I decided not to go fully into it. However, I do need you to know what oxidation and reduction means. And I think what will help you with that is the acronym oil rig, okay? What does oil rig stand for, or what does it represent? Oil rig for oil, for oxidation is losing. Okay, oil for oxidation is losing. Losing what?
Electrons.
Electrons or hydrogens, that's right. And rig is going to be reduction is gaining. Rig is going to be reduction is gaining. Gaining what?
Electrons or hydrogens.
That's right, electrons or hydrogens. Okay. So I'm not going to go into why is something called reducing when it's actually gaining, etc. It has to do with the charges. And really, in the great scheme of things, it's not going to be that big of a deal or important for this class. However, I do want you to, again, understand that redox reactions, oxidation reduction reactions, are always coupled. They occur in pair. There is always going to be an electron donor and an electron acceptor, and that is your redox pair. And we see these type of reactions occurring. In fact, this is going to be important as we go traveling down the electron transport chain. Okay, enough said about that. I want to begin now talking to you about the various steps in carbohydrate catabolism or cellular respiration. So at this point, what I want you to do is I want you to have a separate sheet of paper in which you're going to be taking notes along with me, okay? Completely separate, so get a separate sheet of paper, and this is what we're going to be doing. We're going to be going back and forth with it, and what we're going to do is the following. Gosh, how did I do this again? Oh, here we go. Okay, screen. Okay. Let's see. All right, you see my whiteboard?
Yes.
So this is what I want, thank you. This is what I want you to do. I kind of wrote myself a little cheat sheet, but it doesn't have a whole lot. All right, so I'm gonna start, I'm gonna make a big table, so you need the whole sheet of paper. Yeah? Let me write it down and make sure that I get it correct first. So I want you to have starting compound, oops. Pathway. number of ATP or reduced coenzymes
Wala, Rocky.
or reduced coenzymes produced, okay? And then I want you to tell me the ATP production method. And your options are SLP or oxidative phosphorylation. We just discussed that, right? So what we're going to start with is the first pathway of cellular respiration. That first pathway is going to be glycolysis. Yeah? What is the starting compound for glycolysis?
Glucose.
That's very good. Very good. All right, so we're going to be going back and forth to this. So I'm going to stop right here with this, and I'm going to continue with my lecture. Yeah. Do you see my PowerPoint again? Yes.
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 Okay. Because everything disappeared from me again. Of course it did. There you are. Okay, good. All right. So let's continue. We're going to begin with glycolysis. Glycolysis, remember, means, what does it mean? It means sugar, breaking of sugar, right? Glucose is the most common energy source that's used by cells. I already mentioned that to you, right? And microorganisms actually produce energy. Microorganisms, remember this is a microbiology class, so we talk about microorganisms, specifically prokaryotes. Microorganisms produce energy from glucose via two different processes: cellular respiration and fermentation. But we're going to first discuss this cellular respiration or carbohydrate metabolism, whatever it is that, however it is that you want to discuss it or refer to it. So glycolysis is the oxidation of glucose to pyruvic acid. All right. And it's usually the first stage in carbohydrate metabolism. There are two stages to this. We see that there is the energy investment stage. In the energy investment stage of glycolysis, we're actually, instead of generating ATP, we need to start off by using ATP. So we actually have to use a couple of ATP to get the reaction started. Here is my 6-carbon sugar. Yeah. And during this energy investment stage, we actually break down this 6-carbon sugar into 2 3-carbon molecules. So after this stage, we basically double everything. Yeah. The second stage of glycolysis is going to be the energy conserving stage. And in the energy conserving stage, guess what? We're actually generating ATP, which is energy rich. But you know what else is energy rich? Is this reduced coenzyme called NADH. Okay. So these reduced coenzymes, specifically the NADH, will then travel to the electron transport chain, where through a series of redox reaction or oxidation reduction reactions will generate a large number of ATP. But we're not there yet. So this is just an overview of what's ahead, okay? All right, so let's begin here. As I mentioned to you, glycolysis means splitting of the sugar. This is going to be a 10-step pathway. You do not need to be completely familiar with every single step. It is so interesting how things have evolved over the past decades. When I first took microbiology, we had to know how to draw every single intermediate, not just know the name, but draw it. Now, as a professor, they don't even give you the structure. They just put carbons, like cartoon pictures of balls with the C in there. It's so unfair. It's so unfair. But you know, and I always wondered, why do I need to memorize? Why do I need to know how to draw these structures? When would I ever need it? And then I thought, well, maybe if I can become a professor, then I will. Well, guess what? Look, it's useless, right? So this is actually kind of nice that you don't have to worry about it, even though I was tortured by this. So let's take a closer look at this pathway. As I mentioned, we begin with glucose. Glucose is going to be our most energy-rich sugar. We begin by utilizing a couple of ATP at various different steps. At one point, we actually split the 6-carbon sugar into 2 3-carbon molecules. Yeah, and that is why you see here that we actually are reducing these reduced NADH. There was actually 1 per 3-carbon molecule. Okay, that was reduced, and so that's why we have a net of 2. So when we take a look at this pathway, we see that our starting molecule is glucose. And when it's all said and done, we're going to end up with 2 3-carbon molecules of pyruvic acid, also known as pyruvate. Okay, so our starting compound is 1 molecule of glucose. And our ending compounds are going to be pyruvic acid, actually, to be specific, 2 molecules of pyruvic acid. Why 2? Because remember that somewhere in this cycle, we split this 3-carbon molecule into 2 3-carbon molecules. Yeah? All right. And then we also generate ATP. Wait a minute. Why does it say that our ending compounds is 2 ATP? I actually see here 2 plus 2 equals 4, right? So is that a mistake?
2?
What was that?
Doesn't it use 2 ATP?
That is correct. I do generate a total of 4, but I need to consider the fact that I used 2 ATP at the beginning to get this reaction started. So I must subtract it. Yeah? I must subtract it. All right. So now what I'm going to do is I'm going to go back to my table here. Okay. It's not as convenient as in the classroom, but we do the best we can. Okay, am I back here? All right. Now that we remember, we just looked at it. How many ATP are produced in glycolysis? That's right, Marvin, 2. 2 ATP. Do we produce any reduced coenzymes? Well, let me ask you a question. What do I mean by reduced coenzymes produced? What are my reduced coenzymes? Or what could they be?
NADH.
NADH or?
FADH2.
That is correct. Did we generate any reduced coenzymes in glycolysis?
Yes.
Yeah. What did we produce?
2 NADH.
We generated 2 NADH. And guess what? We know that for every one NADH produced, this will generate 3 ATP in the electron transport chain. 1 NADH will generate 3 ATP, and 1 FADH2 will generate 2 ATP. So if we do the calculation, if we do the math, eventually these two NADH, when we multiply by 3 ATP, they will eventually generate how much ATP?
A lot.
6.
6 ATP in the electron transport chain. So now my next question is, what ATP production method was used to generate my 2 ATP and to generate my 2 NADH? Let's take one at a time. For my 2 ATP, which ATP production method was utilized? You have two choices. Even though there are 3 methods of generating ATP, remember that? We're only going to focus on 2. So your options are, you got a 50% chance, right? Substrate level phosphorylation, also known as SLP, or oxidative phosphorylation. What method was used to generate the 2 ATP directly right there, boom, in that system? Remember that? The way that we generated this ATP is we phosphorylated ADP to give us ATP directly within the glycolysis pathway.
SOP.
Say that again, Adrian.
SOP.
You are correct. Do you understand why? Do you guys understand why? Who doesn't understand why? Raise your hand. Okay. Remember that substrate level phosphorylation. If we go back, is it sharing the PowerPoint?
Not yet.
Okay. Remember that if we go back, not that back, not that far, okay, here. Substrate level phosphorylation means that we're transferring a phosphate to ATP. And as a result, we generate ATP, boom, right there and then, right there in that system. So when I go back to this pathway and I take a closer look at what's happening here, we see that I go from ADP directly to ATP. It doesn't show you the exact process of what's happening, but the only way that you can go from adenosine diphosphate to adenosine triphosphate is if you add a phosphate. And that phosphate was added in the pathway. Yeah? Darlene, did you have a question?
Oh, never mind. No.
Okay, but does that kind of help? Does that make sense? So this is why it's through SLP. Yeah. All right. Let's see. Turn this. All right. So that is through SLP. Now, what about my 2 NADH that was generated? Through what ATP method were these NADH, were these reduced coenzymes generated? Well, let me ask you this.
Reduction.
Is it SLP? Why not?
There's no phosphorylation and it goes to the ETC.
That is right. There is no phosphorylation of ADP. And furthermore, remember, it's in red because these reduced coenzymes had to travel to the electron transport chain. Therefore, the eventual ATP production method from these reduced coenzymes will be oxidative phosphorylation. A P with a circle refers to phosphorylation, by the way. That's the abbreviation. Okay? Now I know what I forgot to add to this. I wanted to add my ending compound. It's okay. Let's see. continue. All right. So it's a 10-step pathway. We know what we start with. We know that our ending compound will be 2 molecules of pyruvate. Function, okay, we generate energy and ATP. We already talked about that through substrate level phosphorylation. It's going to generate reduced coenzymes. How? Through oxidative phosphorylation. We don't see any phosphorylation happening with this NAD. And we don't see any ATP directly produced here with this NADH, correct? We don't. All right. Where does glycolysis take place in a prokaryotic organism? It takes place in the cytoplasm. That is important, okay? And what's important to note is that this pathway does not require oxygen. Why is oxygen such a big deal? For prokaryotes, it is a big deal. Can we live without oxygen? Can we survive without oxygen? No, but we're not prokaryotes. Some prokaryotes can. We can't, but some can. Okay, so questions. Where does glycolysis take place?
Cytoplasm.
In the cytoplasm, that is correct. Glycolysis energy yield will be what?
2 ATP.
2 ATP, that is correct, 2 ATP. Glycolysis breaks glucose into 2 what?
Pyruvic acid.
2 pyruvic acids. 2 molecules of pyruvic acid, also known as pyruvate. They're used interchangeably. Okay, they're used interchangeably. So the next pathway that we have, which for some reason when I was in school, they always seem to skip this little preparatory step and make students kind of guess, well, how do we get from glycolysis to the Krebs cycle? There is an important preparatory step, and it's important for prokaryotes too. But you know, in eukaryotes, it's a little bit more complex, perhaps, right? You see, the problem is that we cannot go straight from glycolysis to the Krebs cycle. And the reason why is because the end product of glycolysis, which is 2 molecules of pyruvic acid, is not the starting molecule for the Krebs cycle. Well, that would be a problem, huh? So there needs to be some sort of transition that occurs. So the preparatory step is often referred to as the transitional step. We're sort of transitioning our end product from glycolysis into something that can actually enter into the Krebs cycle. Okay, so what we see here is the following. We're going to start with our pyruvic acid. We see that pyruvic acid is a 3-carbon molecule. And at the end, we kind of see a carboxyl group here, don't we? When we remove this, we actually remove a carbon dioxide. It's a decarboxylation reaction that occurs. We're removing carbon dioxide, okay? As a result, we're going to end up with a 2-carbon molecule. This 2-carbon molecule, known as acetate, is going to bind with coenzyme A to give me a final product of this molecule referred to as acetyl-CoA or acetyl-CoA. You say tomato, I say tomato. Okay. So this acetyl-CoA is now the starting molecule for the Krebs cycle. So we start with our pyruvic acid. We have a decarboxylation reaction occurring. We have this acetate bind to a coenzyme A, and in the process, what do we see that we release? What do we generate?
Acetyl-CoA.
Well, acetyl-CoA is our final product. But do we
NADH.
generate, say that again?
NADH.
NADH. Now that's important, right? Because NADH will eventually lead to what? ATP. That's right, ATP. Okay, so let's take a look at this. Go. Now let's talk about our next step, which is going to be the preparatory step. Oh, that's not where I meant to write it. What is our starting molecule for the preparatory step?
Pyruvate.
To be specific, it's going to be 2 molecules, right, of pyruvate. Gotta be specific, yeah? How do we generate any ATP or reduce coenzymes within this preparatory step?
Yes.
Yes? What do we generate and how many?
NADH.
We generate NADH. How many?
Is it 1 per pyruvate?
That is correct. It's 1 per pyruvate. Therefore, we will generate how many?
2.
2.
So we generate 2 NADH. And I know that 2 NADH times 3 ATP will equal eventually generation of 6 ATP, correct? Now, through what ATP production method was this NADH generated? Huh?
Oxidative phosphorylation.
Oxidative relation. Are we getting this? Are you guys getting it? A little bit, hopefully.
Professor,
Yes.
for this step, do you want us to know all the acetate being added and all the other steps, or do you just want to know?
I want you to know the starting compound, and I want you to know the end compound, and I want you to know any reduced coenzymes or ATP produced and through what method.
Okay, thank you.
Yeah, I think the overall reaction, you'll know that there is some carbon dioxide. I don't need you to know that the coenzyme A necessarily comes in and binds. So now what we have here is we have acetyl-CoA. The acetyl-CoA is what's actually going to enter into the Krebs cycle. How many acetyl-CoAs do we produce? Do we generate?
2.
That is correct, 2. Because you generate 1 per pyruvic acid, right? Therefore, you're going to generate 2. And so now we have each of these acetyl-CoAs will enter into the Krebs cycle 1 at a time. And they go through this cyclic pathway. This is an 8-step cyclic pathway. There are 2 turns per molecule of glucose. Why?
Because each one enters 1 at a time?
Because glucose, yeah, but because if you think about it from the beginning, the reason that we have 2 turns per 1 molecule of glucose, so the question is, we start with 1 glucose, why are we going around it twice? Because glucose gets broken down into 2 pyruvic acid, which then gets converted to 2 acetyl-CoA, which then each acetyl-CoA enters the pathway, yeah? So take a look at the pathway. You do not need to know all these intermediates, and you definitely don't need to know how to draw the structure, okay? If this was my molecular class, you would need to know those intermediates, but not necessarily draw them, but you would need to know them, but not for this class. There's a lot of detail involved in this, a lot of detail. What you do need to know is your starting compound. If you generate any ATP or any reduced coenzymes, And what, that is what you need to know. So let's take a look at this. Do we see any ATP generated in this cycle? Say
Yes.
it again. Okay. Yes?
Yeah.
How many ATP do you produce through the Krebs cycle per 1 molecule of glucose?
1.
1? Are you sure? 2? It's going to be 2. Remember that we're going to have 2 acetyl-CoAs going into the cycle. Yes?
Yes.
All right. Do you see any reduced coenzymes generated? Yeah?
Yes.
Yeah, Kelly says yes. Which ones?
NADH.
Okay, let's start with NADH. How many NADH do we generate per molecule of glucose?
6.
6? Well, let's see. I see here 1, 2, 3. I only see 3.
So 3.
Yeah, but 2 turns per
That's
molecule.
right. We have 2 acetyl-CoAs that go in. Therefore, we're going to generate a total of 6 NADH. Do we generate any other reduced coenzymes?
FADH2.
FADH2. How many FADH2 do we generate per 1 molecule of glucose?
2.
That is correct. We generate 2. So now let's go back to my whiteboard. Oh, no, I did the wrong one. Am I sharing my whiteboard? No?
No.
No, okay, I clicked on the wrong one, sorry. There we go. Am I sharing it now?
Yes.
Yeah.
Okay, all right, so let's take a look at my whiteboard now. And I'm going to write the Krebs cycle here. And now you guys are all gone. You disappeared on me. Oh, well, it's okay. Krebs cycle. What is my starting molecule for the Krebs cycle? My starting compound?
Acetyl-CoA.
Acetyl-CoA.
2 molecules, right? Of acetyl-CoA. All right. Did we produce any ATP? You said yes. We generated how many ATP total?
1 per acetyl-CoA.
For a total of?
2.
Okay. Did we generate any reduced coenzymes?
Yes.
And we said that we generated
6 NADH and 2 FADH.
H.
6 NADH, which we know 6 NADH, you have to do the math, right? Times 3 ATP equals 18 ATP. And we also generate how many FADH2 did you say?
2.
2.
2, FADH2. And we know that we have 2 FADH2 times 3 ATP?
2.
2. This one generates less ATP, guys. And I'll show you why in just a bit. Times 2 at ATP equals a total of 4 ATP that will be generated. Now let's go back here. What is the ATP generation method for the 2 ATP that were generated? What is the ATP production method? Is it SLP or oxidative phosphorylation?
SLP.
SLP.
And what about the NADH? What method is it going to use to generate ATP?
Oxidative phosphorylation.
Right? Oxidative phosphorylation. And what about the FADH2?
Oxidative
Oxidative
That is
phosphorylation.
correct. Oxidative phosphorylation. And there you have it. You need to understand this. You need to understand it. Oh, how did you guys get here? Do you see my screen? Is it normal or is it weird?
No,
it's
that's
the-
a weird one.
It's normal?
It's the
No.
presenter
No.
screen.
It's
Retard.
not normal. Okay, I knew it. I knew something weird was happening. Hold on. Is it better now?
Yes.
Yes.
Yeah.
Yeah, okay. Is it still normal or no?
Yes.
Okay, so our starting compound is acetyl-CoA, as I mentioned, and our ending compound, you don't need to know about oxaloacetic acid necessarily, but there is carbon dioxide generated, and there is NADH, FADH2. Okay, so the function of the Krebs cycle is to generate, reduced coenzymes. This is why it's hard to teach remote. It really is hard to teach live remote. At least when I record it, I can edit all this stuff. I don't have to worry about it. It generates reduced coenzymes, NADH and FADH2, as well as ATP. And now you understand the methods through which these were generated. I hope, yeah? So make sure that you review this because it may make sense now, but if you do not review it, you will forget it, okay? So please make sure that you review this. Let's continue on. All right, so the Krebs cycle, the location is important. In prokaryotes, the Krebs cycle occurs in the cytoplasm. You know what I forgot to do, which was very important? What is my total ATP yield? What is it? You need to write that. I don't want to go back and forth because it keeps on messing it up. So that's what I would write. Total ATP. Well, let me do it actually.
Like 34, isn't it?
34. Yeah, let me go back to this. It's okay. We can just continue to go back and forth. That's okay. Hopefully I won't have that problem anymore. Let's see. Nope, it's not working. Let me try it again.
32.
32. Well, let's see. Let's investigate, guys, because I'm glad that I remembered this question.
Maybe 38.
We have 2 plus 6. Yeah, that's 8,
Like
10, 16. Yes. What is this? 34
Before.
plus 4, 38.
38.
All right. Let me ask you a question.
Wait, Professor?
Wait, you forgot the 2 ATP.
Hold on a second. Yeah.
From the Krebs.
It's still 38. I just counted in my head, but I forgot to circle it. But let's count it. Let's add them. So 2 plus 6 is 8, right? 10, 16. Yes. 16 plus 20, 36. Whoa, did I do it wrong? Now this doesn't make sense.
You circled NADH instead of, yeah, you just circled an extra one.
I circled an extra one. I circled the wrong one.
The prep step.
Not good. There we go. Am I correct now? Shall we do it again? 2 plus 6 is 8. Oh, that's what I was doing. I was saying 10. It's a long day. 8 plus 8 here is 16. 16 plus 18 is what?
34.
34.
34 plus 4 more is 38. Oh, I misspelled prokaryotes. What about eukaryotes? How many, what's the total ATP that we produce?
Isn't it 30 to 32?
30 to 32, it's usually about, let's say about 36 ATP. 34 to 36 ATP usually. Oh my gosh, wait a minute. Am I saying that bacteria are more energy efficient? That they produce more ATP than us that are more sophisticated? Is that possible? Are you surprised? I'm shocked. I mean, I think that I would require more energy than a single cellular organism, right? What do you think? Why? Did I make a mistake in my calculations? Initially I did, but no. Why are prokaryotes able to generate more ATP than eukaryotes? Interesting. We don't know. It has to do with the location of the Krebs cycle. Okay, the Krebs cycle in prokaryotes occurs where?
Cytoplasm.
In the cytoplasm. Whereas in eukaryotes, the Krebs cycle takes place in the mitochondria. And remember, we talked about how do we transport things through plasma membranes and stuff like that. Mitochondria has a plasma membrane. And the way that the acetyl-CoA or even the preparatory step, in order to get the pyruvate in, it requires ATP. The preparatory step occurs in the inner membrane of the mitochondria, and it requires ATP. And how many ATP does it require? It requires 1 per molecule. It requires 1 per pyruvate. So right off the bat, we'll have at least 2 less, okay? That's why. Now the final electron acceptor, when we generate all this ATP from these oxidation reduction reactions of our reduced coenzymes NADH and FADH2 is going to be
Oxygen.
oxygen. It will be oxygen. That is correct. Very good. All right, let's continue on. So we see here that the location of the Krebs cycle is going to be the cytoplasm. And now you know why that's important, right? Now you know why they generate more ATP than us, sophisticated eukaryotes, or so we thought. Whereas in eukaryotes, it occurs in the mitochondria. Oxygen requirement, it does require oxygen. Okay. Types of reactions that occur in the Krebs cycle. We see oxidation reduction reactions, which is going to generate what?
The coenzymes.
The coenzymes, NADH and FADH2. We see decarboxylation occurring in the Krebs cycle, which is going to release
CO2.
that's right, carbon dioxide. And substrate phosphorylation or substrate level phosphorylation is going to form or generate what?
ATP.
That is correct, ATP. Okay. All right, so now let's finally talk about the electron transfer chain. I hope I have enough time to finish this. Yes.
What was the first one? Oxidation reduction, which generates what?
The reduced coenzymes NADH and FADH2. I'm going to have to speed it up a bit because I'm running out of time. All right, so the electron transport and oxidative phosphorylation. Those reduced coenzymes that were generated throughout the various pathways of glycolysis, the preparatory step, and the Krebs cycle, those reduced coenzymes, which are NADH and FADH2, guess what happens? They all travel to the electron transport chain. So the function of the ETC is to oxidize those reduced coenzymes that were produced in those various pathways to produce ATP. And now you know at what ratio, right? 3 ATP per NADH, 2 ATP per FADH2. Now the mechanism in which this will be generated, the mechanism in which ATP will be produced is chemiosmosis. Okay, energy is harvested from these oxidative phosphorylation reactions. And I'm going to show you how that happens, sort of, okay? The location of the electron transport chain is important. It is in the cell membrane, also known as the plasma membrane in prokaryotes. In eukaryotes, it's going to occur in the mitochondrial membrane. Well, why does it occur in the mitochondrial membrane for prokaryotes, guys?
Because the Krebs cycle
RNA.
takes place in the mitochondria.
Yeah, but why doesn't that occur for prokaryotes? Why doesn't it occur in the mitochondria in prokaryotes?
They don't have
We don't have
mitochondria.
one.
That's right, George. They do not have mitochondria. They do not have organelles. So oxygen requirement, there is an oxygen requirement for the electron transport chain. So here is our starting compounds for the electron transport chain. When we total up all the reduced coenzymes that we've generated in the various pathways, we end up with 10 NADH and 2 FADH2. At the end of the electron transport chain, guess what we're going to end up with? The NADs and the FADs. Okay. So here we see the electron transport chain. We see that NADH is going to enter the electron transport chain and go through a series of oxidation reduction reactions. As it goes from protein to protein being reduced and oxidized, oxidized and reduced, etc., it is releasing protons. It's releasing hydrogens. One of the reasons that NADH generates more ATP per NADH compared to FADH2 is because NADH enters the Krebs cycle earlier. If you notice, you'll see that FADH2 enters the Krebs cycle at a later time. Yeah. Therefore, it's going to go through arguably less oxidation reduction reactions. As a result, it will generate less protons. Yeah. So what happens is you create a gradient and you have many protons or hydrogen ions on the outer compartment compared to the inner compartment. Yeah. And so what happens is a process referred to as chemiosmosis. The electron transport chain produces ATP by chemiosmosis. This involves a system referred to as ATP synthase. There is a proton motive force that as these protons are pumped through this ATP synthase, this nodule here, it's kind of like a knob, it rotates with such a force that it phosphorylates ADP to generate ATP. The mechanism in which this happens is actually a lot more complicated than I'm stating it. In my molecular class, I actually have students read the journal article where this process was first described. It is very fascinating, but it is beyond the scope of this class. In fact, the figure in your book doesn't even show the knob that actually holds this top part stationary with this knob. So it rotates, but it's also held together, creating this friction. And when it releases, because it's kind of held together by a knob, it over rotates, so to speak. So I can't even explain it because your book doesn't have the accuracy in the figure. So what you should know is what basically the definition and the purpose of chemiosmosis. Okay. And that is electron transport carriers. Carriers are going to shuttle electrons as they actively pump hydrogen ions. You see them pumping hydrogen ions, also known as protons, across the membrane, setting up a gradient of hydrogen ions referred to as a proton motive force. This proton motive force is going to generate the vast number of ATP. The final electron acceptor, when it's all said and done, as these reduced coenzymes go through a series of protons being oxidized, reduced, that final electron will be accepted by oxygen, which will bond to a couple hydrogens to generate water. In other words, we say that the final electron acceptor is water. This is another cartoon picture of what these proteins look like embedded in the plasma membrane. For this class, you do not need to know the names of these proteins that are embedded in the membrane, okay? Ubiquinol and all that, you don't need to know them. Aerobic respiration, as a review, converts glucose to carbon dioxide and water and releases energy in the process. The overall reaction is C2H12O6 plus 6 hydrogens, which will yield carbon dioxide plus water. It's interesting because the reverse reaction along with sunlight is photosynthesis, but we don't talk about photosynthesis in this class, okay? Glycolysis plus a transitional step, also known as a preparatory step, and the Krebs cycle plus the electron transfer chain all encompass aerobic respiration. And as you may remember, oxygen is the final electron acceptor. This is not from your book, but it just kind of gives you, again, the yields of glycolysis, et cetera, et cetera. This is also not from your book. I don't believe it is, maybe it is, but this is just a nice overview of the different ATP generation methods, okay? And I like this because it gives you a visual of glycolysis and that it produces ATP, but it also shows you the reduced coenzymes that are generated in each step. And then you notice that all these reduced coenzymes that are generated, they're all purple. They all travel to the electron transport chain. So it's kind of nice that it shows you that. All right. And in the process, we're going to generate in the electron transport chain 34 ATP. It's just awesome, right? This one is the one that's not from your book, but I kind of like it. It's a nice little overview. And again, it's basically more of the same thing, But we need to quickly go over fermentation before my time runs out. So fermentation, we know that it begins with glycolysis. Yeah. The function is to regenerate. This is important. Why do we have fermentation? Well, first of all, not every microorganism can exist or survive in the presence of oxygen. And we're going to learn more about that in the lecture. So the function of fermentation is twofold. Regenerate oxidized NAD+. That's going to be important. I'll show you why. Regenerate oxidized NAD+ and produce ATP. But it's not going to be as rich in producing ATP as cellular respiration. It's not. Where does this happen? In the cytoplasm. Remember that in the cytoplasm, no oxygen is required. So glycolysis occurs in the cytoplasm. Yeah? All right. No oxygen is required. The starting compound for fermentation is going to be pyruvic acid. Why is it pyruvic acid? Well, because that is our end product from glycolysis. But am I being specific here? What should it really say here that my starting compound for fermentation is? What would be more accurate?
2 molecules of
Thank you. That is correct. 2 molecules of pyruvic acid or 2 molecules of pyruvate. Because remember, my starting pathway is glycolysis. After glycolysis, I take my 2 pyruvate and I take them through fermentation. My ending compounds for fermentation will be different depending on what the product will be. It could be alcohol, it could be an organic acid. Okay, so here we see fermentation, we see glucose, we generate, in glycolysis, we generate ATP, right? We generate 2 molecules of ATP, correct? But we also generated this reduced coenzyme NADH. If we want to go through glycolysis again, we need NAD plus. So part of the fermentation process is to regenerate this NAD plus. You see that? So that it can be used again in glycolysis. So when we're looking at fermentation, produces this reduced coenzyme NADH. The only way we're going to generate NADH is if we have NAD plus. Therefore, in fermentation, we need to regenerate NAD plus so that we can have further eventual fermentation. Now, in glycolysis, our end product is 2 molecules of pyruvic acid. Pyruvic acid can then take different paths. Pyruvic acid can be decarboxylated and further processed to generate ethyl alcohol. No oxygen present. This is why when you make alcohol, beer, wine, it's usually under anaerobic conditions in order for fermentation to occur. Or we can take our final molecule here, pyruvic acid, 2 molecules of it, and we can further process it to generate lactic acid. Now that actually burns, right? When you're running and you're running it so much, you exhaust your muscles and you generate this lactic acid under anaerobic conditions. But in either process, we see that we are regenerating this NAD plus because it's needed in glycolysis. I hope you understand that. I hope you understand that. There are many different end products for fermentation. You don't need to know them all. You should just know the ones that I mentioned. Alcohol production, ethanol, and lactic acid production. But here we see that after glycolysis, if our starting molecule is pyruvate for fermentation, which it is, right? We can have many different types of end products depending on the microorganism that we're talking about. Propionibacterium is called propionibacterium because the end product is propionic acid, et cetera, et cetera. Again, you don't need to know these, yeah? So overall, fermentation, 2 examples. Alcoholic fermentation, we see this occurring in the yeast Saccharomyces, where we go from pyruvate that was generated in, oh my gosh, I forgot the pathway that this pyruvate is generated in. Help me out.
Glycolysis.
Glycolysis. And the pyruvate is going to enter fermentation phase to generate ethanol and carbon dioxide. Or lactic acid fermentation, where we start with our 2 molecules of pyruvate and our end product will be lactic acid. Lactobacillus. Lactobacillus is good in yogurt. It's good for good gut health. So when we're talking about fermentation, how much ATP then does the cell produce with fermentation?
For ATP.
4 ATP, Jake? Is he correct?
Is it 2?
It is 2. The only ATP that is generated with fermentation is the ATP that was generated in glycolysis. And although you're right, they do produce 4 ATP, Jake. Remember, we must subtract the 2 ATP that are required to start the process of glycolysis.
It's Gerardo.
Oh, Gerard, okay, sorry, I thought it was Jake. Okay, so this is a nice little table that tells you some of the commercial applications of fermentation. Microbiologists take advantage of this in various food production and in experimenting and testing. So microbiologists would work on this. I mean, if you decide to go into pathomicrobiology, one of the classes that you'll take is food microbiology, and you get to make a lot of cool stuff using various culture. And then nobody wants to eat it because they're kind of worried about what you put in there. So, you know, anyways. And finally, here we see that there are other sources that can be used for glycolysis, but we know that glucose is by far the most used and the most energy rich. Okay, so